And without further ado, we will continue to Luis. Luis, are you here already? The third speaker? Okay, thank you.
Can you hear me?
Yes, we can hear you clearly. So Luis, Filipa, okay.
Thank you.
Okay, please, the time is yours, Luis. You have 15 minutes.
Okay, I'm measuring my time here. So, I'm Luis Gonzalez. I'm a current PhD student of genomics at the University of Cambridge. And the work I'm about to present is actually what was my MPhil work. I'm mostly working to present new to bioinformatics tools that actually are based for the microbiome study. My work is titled In Science of Phylogenetic Trends in Early Life Bifidobacterium breve. So, as we already know, the early life microbiome is actually accomplish an important role on health and disease. We already know in the case of neonates, just infants that were recently born less than 1 month of life, the microbiome actually accomplish a really important role. For instance, it participates in the maturation of the immune system and enterocytes, accomplish a really important factor there, and also is playing an important role in the protection against infections. Especially, they have been recently found that in newborn babies with the healthy microbiome, actually there were more 3 prevalent species that were found. Both, all of them were bifidobacterium, but especially the species breve, legum, and bifidum. And actually, in this case, I'm mostly going to talk about breve as actually a really important bacteria, which is it's the one that actually the first colonized the infant gut microbiome and make sure, to create an anaerobic environment there in order to favorite the colonization of all the probiotic bacteria that might, come later and then participate in the maturation of the gut on infants. And actually, there has been also found that administration of breve has clinical outcome of reducing the necrotizing enterocolitis in infants, and also prevention of rotavirus-induced diarrhea. Despite actually being breve an important probiotic that has been reported, actually its phylogeny has not been totally explored yet. Actually, those were the most recent studies that were published that on 2014 and 2020 that actually use only 17 or 79 genomes. So actually these are so actually here's where I came. That was the aim of my study. So actually I proposed me to understand breve's phylogenetic structure and how it relates to host geographic location and age. But the difference between previous studies is that I was not aiming only to using isolates. In my case, I want to reconstruct genomes based on metagenomic samples. And in this case, from what is called metagenomic assembled genomes, MAGs. Basically, a MAG basically consists taking a sample, in this case, a sample that contains harbors multiple bacteria. Then we could perform a shotgun metagenomic sequencing, and knowing the coverage of the specific the member that we want and the sequence characteristics, we will be able to, based on those shotgun sequencing, we can split individual genomes of this shotgun metagenomic sample, and then, look at individual genome of bacteria, even if we have not any culture at all. So in my case, actually, that's the strategy I follow. You combine isolate genomes plus metagenomic assembled genomes in order to understand more about the phylogenetic structure of breve. So here, the latest study, the most recent study has only 80 genomes. In my case, I managed to build 183 genomes, which actually is a really huge increase there. After applying a quality threshold of 90% of completeness and less than 5% of contamination, actually end up having 775 high quality genomes, of which 623 were actually MAGs and 152 were isolates. My case in my lab currently at Wilkins Sanger Institute, my lab were actually currently working on the baby biome study, which actually studied that focused on looking to the neonate microbiome in infants in the UK. Most of my samples also most of my breve also came from that study, more than the half of those MAGs. And the other half came from Magnify, which is actually a public database from the EBI for MAGs genomes. Regarding isolates, most of them came from the NCBI. They are publicly available. On the second place, we have some private genomes that we have on the PBS. They are about to be released. And then we have also genome from GCM and IMG databases. Now, in my case, I really want to analyze the structure of this phylogenetic tree of breve and relate this to geographic location and the age group of the host. Talking about the geographic location, most of my genomes came from Europe because of the BBS study, and most of the genomes available came from Europe. In second place, we have Asia, North America, South America, and Africa. In this case, sadly, regarding South America and Africa, we only got like 3 genomes for each continent, so we would not analyze those on my future slides. Regarding the age distribution, most of my samples came from neonates because, again, most of my samples came from the BBS study. Infant on second place with 241, and adults at the end. So the phylogenetic analysis workflow I actually follow is, once I already collect all my genomes, I actually make an annotation, in this case using POCA. Then in my case, I calculate the pen genome using Rowry. And then what I've done was making a core genome alignment. So basically, I'm aiming for a tree that is basically contain the functional properties of breve. And then in my case, I use RoxML to build my phylogenetic tree, and then FastMap in order to identify all the possible clusters that actually I identified in my tree, and then relate this with the information of the host. So the first result that I actually had was that breve actually possess an open pen genome. And actually what that means is that if we actually have an hypothetical scenario in which we build a graph, on the x-axis, we look at the number of genomes we actually have in our database, and in the y-axis, we look to the pen genome size. We will see, for instance, there are some bacteria that no matter how much genome we actually add to our database, if we look to the number of genes of those entire database, the pen genome size, we will see that despite we add more genomes, we will not see any new genes at all, because there are species that actually don't participate at all on the horizontal gene transfer process. Or for instance, on the other case, we have E. coli, which actually is really famous for acquiring new genes because it performs active horizontal gene transfer process. And we will see that if we add more E. coli to our database, we will find always new genes because it has an open pen genome. So in the case of breve, actually, there were a lot of studies that claim that breve has closed pen genome that actually don't participate in many horizontal gene transfer process at all. But actually, here I look after adding my isolates and my max, actually, that I was finding a lot of genes, and actually this has the form of an open pen genome. So this is actually the first time we actually realized that breve might participate actively in an horizontal gene transfer process, and might actively participate, exchanging genes with the microbiome in this environment. So actually, this was something really cool to find. Now, looking to the phylogenetic tree, in this case, is a tree based on functionality. We have seen here 2 clades, the C1 and C2, and the external ring, you can see actually the colors in sky blue that actually my genomes that are max, and in red, the genomes that actually are isolates. And in the internal ring, you can see all the subclades that actually I have found for each clade here. So for instance, for the clade 1, I have actually looked for 4 new subclades. And for this clade 2, I have found like 6 new subclades. That actually, this was super interesting. I want to put your attention here that actually, in this case, all these new clades here, this one and this one, actually composed entirely by max. So actually, this has been really cool, since actually that showed us using max, we were able to uncover new subclades that were not so evident before. So actually, this was something really cool to find. And also here, there were some clades that already existed here by looking only to the isolates. But just the fact of adding more max to your phylogenetic tree actually, makes more evident the existence of those clades, and I was able to identify them. So actually, max helps to uncover C1 subclades and confirm the existence of other ones. Now, when I actually look to the information regarding my phylogenetic tree and information of the age of the host, in this case, neonates, infant, and adults, I realized that neonates and infant actually were really rich on genomes that came from the clade C2. And actually, it was super interesting because even we're talking about the same species, that is Bifidobacterium breve, but it's super interesting that actually one specific clade has a preference to a specific host. In this case, infants and neonates. And remember, this is a tree that actually was based on functionality. So actually, this actually is really interesting. And I was thinking, actually, there's a literature that says that Bifidobacterium breve that actually came from an infant actually has genes that actually are related to digestion of sugars related to milk, which actually makes sense since breast milk is the main part of the diet of infants and neonates, compared to an adult that actually don't have any difference at all. So that was something really interesting to find. Regarding the geographic information of these patients, in this case, I had found that Europe and North America genomes actually also reach on the C2 clade of breve. And when we look to the Asian population, actually those are actually enriched on the clade C1. And actually those differences were not evident until the addition of MAX. So actually this was super interesting. And again, I'm thinking again, these clades are based on functionality. So we're thinking that maybe some clades has any preference to a specific host just because of the lifestyle of each host, and it might encode different things. And actually that was super interesting to find, because like even we're talking about the same species, but there are some specific clades that has preference to a specific host. So that's actually, this is the first step, you know, to make personalized treatment. You might find the same Bifidobacterium breve, but this same breve will not be the same that actually is present in a host that might be a South American population versus an Asian population, because we already have different lifestyles. So we will actually uncover which clades are actually more specific to each host according to its geographic information or to its age information. We will be able eventually to build probiotic treatments that are specific to the information of this host. So actually, this is the first step for personalized treatments using probiotics. So some key messages actually to take home is that actually the inclusion of MAX in breve database allow to have a better understanding about how actually it's breve's pangenome, which I realized that breve has an open pangenome rather than a closed pangenome. It's like the first time that actually we realized that breve might interact in an active or resultant gene transfer process with its microbiome community. And also we help to discover new clades and subclades. And also we realized that breve clades are associated with host metadata. In this case, we realized that there were some specific clades that were related to infants and neonates, and also that one specific clades that were, in this case, more related to Western lifestyles and region Europe and North America, and one specific clade that was associated with Asia. The main limitation is, again, as I already mentioned when I started my presentation, is that actually we have a really unequal distribution of breve genomes across host geographic locations. And actually, this is a common problem that we have across most metagenomic studies. Most of them are actually focused on Europe and North America. We don't know anything about South America, Africa, or Asia. So actually, fortunately, encouragingly, there are new projects that actually aim to perform metagenomic studies in heavy undersampled regions. In this case, we have Latin Biota that actually its aim actually to collect microbiome samples and analyze them in population of South America. We also have the CHAIN project, the Childhood Illness and Nutrition Networks, which one of its aim is actually to make the largest collection of microbiomes cohort in Africa and South Asia, in order to have more insights about the microbiome population on those regions. Well, finally, I would like to acknowledge, thanks Triple Oli. In this case, this is my current PI here at Wellkin Sanger Institute, participating in organizing this project entirely remotely. Thanks to Jan Xiao, this guy. In this case, this is a postdoc that helped me a lot to organize my thoughts. I'm Nitin, that helped me a lot to realize all my phylogenetic studies. And thanks again, the Wellkin Sanger Institute for funding my info program and the project in general. So that's everything for me. I will be super happy to answer all questions on the next section. Thank you.
Thank you, Luis. Let's give a big applause for Luis. So this is the last speaker in the first session.